nice to come from the basic science side, who tries to put some applied aspects in the research, but then to also hear from stakeholders, from people like you who are affected by the disease, and to see what your view of things are, what the real challenges are in the real world. So as Harry mentioned, I'm not only going to talk about CWD prion detection in fecal samples, but I'd like to give a more broader overview about the research that's going on in my lab and bring it into context what has been found by others. So you might find some of the things are not very much related to the real world application, but I hope I can get the message across of some things that might be relevant for you as well. So, as Catherine already did very nicely, I also wanted to recapitulate what prion diseases are, and I know you are all aware of prion diseases can occur in all mammalian species. So we have it in humans, like the Creutzfeldt-Jakob disease. It's a sporadic disease, and no one knows why some people are hit by the disease. We have genetic forms in humans, and we also have infectious forms in humans, like Kuro, which was a quite curious disease that was transmitted by cannibalism. Then we have prion disease in sheep, scrapie, which is the animal disease that has been known for the longest time, actually since the 14th century already. And then, of course, mad-cow disease, or BSE, that caused a huge crisis in the 1990s and finally was transmitted to humans as well, and caused a new human prion disease that affected very young people, teenagers. And of course, what we are here to talk about is chronic wasting disease in cervids today. However, all prion diseases, although they come in different species, they come they come for different reasons, they all have several things in common. So first of all, they are all fatal, they are all transmissible or infectious, and they can be transmitted between the same species, and they can also be transmitted between different species. However, this is not a random process, so it's dictated by several things. So all prion diseases come with what we call spongiform neurodegeneration, so brain cells die and then leave such holes in the brain, which lead to that sponge-like appearance in the brain. They all have a very long incubation time, which can be like decades in humans, but once the clinical phase starts, so the affected individual starts to develop symptoms, they would die very quickly. So the clinical phase is very short, which also makes it challenging to treat the disease, because at the point the disease starts, it's already gone very far in the brain, and a lot of the brain has been destroyed already. So there's no preclinical diagnosis, and there's no treatment available, and of course all those diseases are caused by prions, however by different types of prions. So prions, Catherine mentioned it already, it's a proteinaceous infectious particle, so it's only protein. It does not have any genetic information. So they're not viruses, they're not bacteria, so they are proteins, and this is what they look like if we purify them from infected brains. So they are those like fibrous protein assemblies. And actually where they come from, it's a little bit like Dr. Jekyll and Mr. Hyde. So on the one hand, every one of us and every mammal expresses the normal prion protein, mostly in the brain. It is a normal protein of our body. It has a function there, although it's not very well characterized, and it does not cause disease. And then we have the Mr. Hyde, which is called PRP scrapie or prions, and this is a molecule that changed its three-dimensional structure, it changed its shape, which makes it then a very sticky molecule that can clump together in the brain and cannot be digested. So once it's in the brain, it builds up such aggregates. It cannot be, so the body cannot get rid of it, and it's the infectious form of the protein. So PRP scrapie is infectious. So what we're thinking about, of course, chronic wasting disease, and question is, such animals, they look healthy, do they have the disease? And actually chronic wasting disease can come, just like an animal that looks healthy like this can have chronic wasting disease, because again the incubation period is very long, and the symptoms start very late. And once the symptoms start, animals start to look like this. They get wasting, they start to drool, they separate from the herds and so on. They develop symptoms and finally they die. So CWD, we've heard a lot about this, so I can go through it quickly. It affects elk, mule deer, white-tailed deer, moose, reindeer. It's in both wild living and farmed animals, which is a huge part of the problem. It's in wild animals, so it's completely uncontrollable, unlike DSE, which was only in domestic animals, so it could be controlled somehow, but once you have it in wild animals, the problem becomes way more challenging. So it's transmitted between animals horizontally, it's taken up orally from the soil, and it's thought to be the most contagious prion disease. Some important questions that are remaining: is there a zoonotic potential? So can CWD be transmitted to humans? So far there's no evidence for this, but Catherine talked about those preliminary results from the macaque studies, which show that there might be potential of transmission to non-human primates. And the other question: is there a potential for transmission to non-cervids, like other livestock, sheep, cattle, and so on? So why is CWD so infectious? Again, as opposed to the classical DSE, which was where we found prions mainly in the brain, this is very different in CWD. So basically, CWD-infected animals have prions everywhere. They have it in the muscle, they have it in the antler velvet, they shed it in saliva, urine, feces, and this leads to contamination of the environment. So the prions can bind to soil. They are very resistant to any exogenous inactivation, so they persist in the soil, and finally this leads to the infection of other animals. So a deceased animal sheds prions in urine, feces, finally they die, the carcass decomposes in the environment, the prions bind to the soil, and then a healthy deer comes and feeds on that ground and gets the disease. So again, I don't want to go a lot into detail. You all know it's in 24 states of the United States and in Saskatchewan and Alberta. And about 2 years ago, CWD was detected very surprisingly in Norway, in Europe, in a reindeer. So this was the first case of CWD in Europe, and it was also the first case in a wild reindeer. Although we knew before that CWD can be transmitted to reindeer, so this was really the confirmation that wild reindeer can become infected with CWD. So of course the Norwegian government, they were very concerned about this, and they decided to shoot or put down an entire reindeer population of about 2,500 animals. So they started the shooting last year, and they test every animal from that herd, and also increased their surveillance efforts in other areas. And so the last I heard was last November, and at that time they found 7 reindeer infected, so it was corresponding to a prevalence of about 1 to 2%. They found infected moose in a different area, and they found infected red deer, positive red deer. Although no one knows so far how CWD got into the reindeer in Norway. And what is also interesting, that the reindeer that were infected, they seem to resemble very much the disease that we see here in North America in deer. So from the distribution in the brain and from how the prions look like, it looks a lot like the North American disease. However, the moose and the red deer, they were different. So for example, these were old animals. They didn't have a lot of infectivity outside of the brain. So people speculate whether there might be a sporadic form of CWD as well, but this has not been confirmed. And of course, it will always be hard to confirm this, because how would you say this is a sporadic disease? But there are, obviously, it looks like there were different types of CWD around in Norway. So the challenges about CWD, of course, it's a highly contagious disease. It's 100% fatal. We have a long preclinical disease phase. It is expanding. The disease range is expanding. The number of animals is increasing, so the prevalence is increasing. We have a persistence in the environment, in the soil. The diagnosis is difficult because we don't have any genetic information, so we're looking just at a protein that changed its shape, so we have to manipulate the test samples to make it detectable. And of course, there's no treatment or prevention method for CWD. So now you might think, what I told you, we have the good prion protein, we have the bad prion protein. That's a simple story because it's one protein which can do its function and it can misfold into that prion conformation. However, it's unfortunately it's not as easy as it looks like. So first we have prion disease in humans and animals, and in both humans and animals, prion diseases are very diverse. So as I said, in humans we have sporadic disease, we have genetic disease, we have transmitted infectiously acquired disease. In animals we have CWD in cervids, BSE in cattle, scrapie in sheep, and all this comes along in humans with multiple subtypes of the disease with different clinical symptoms, whether you have sporadic CJD or a genetic form of prion disease. This can come along with different symptoms with a different distribution of the prions in the brain. In CWD, so Catherine already touched on the prion strains, and I'm going to talk more about this in the following slides, but in CWD it looks like there are different strains and we do not completely understand what that might mean for us. And also in BSE and scrapie, in both diseases there are classical and atypical forms, which also indicates that there are, in all the prion diseases, in all the species we have different prion strains. And basically what a strain means, it's a variation in the 3D shape or in the shape of the infectious prions. And why this is important is because strains can affect the transmission or the transmissibility of the prions from one species to the other. It can affect the diagnosis of the disease. For example, in scrapie, we have the classical scrapie form where the animals start itching and they rub their heads. And in this disease, a lot of the prion infectivity is found in the obex, which is the material that's used for diagnosis. But then there are also the atypical forms of scrapie. They have different clinical symptoms. They don't scratch, they just become ataxic. And in these animals, the prions are distributed differently in the brain, so there's only little found in the obex and a lot in the other parts of the brain. But if you now have such an animal and you take the obex for diagnostics, it might be possible that it escapes if you have a different strain. So that's why I said it affects diagnosis. For transmission, one example is BSE, so the classical BSE, it has been transmitted to humans. And the classical BSE is the one that was caused by the meat and bone meal, so that was transmitted by feeding the animals with contaminated feed. But then there are also atypical forms of BSE. Again, all the animals are affected, distribution in the brain is different, and whereas the classical BSE was transmitted to humans, for the atypical forms this is less clear, so it seems they have less zoonotic potential. So this is what I mean if I say strains can affect the transmission properties. And the strain can also affect whether a certain drug or a treatment that so far only worked experimentally would work against the different strains. So in CWD, there is only limited information about CWD strains. So the first one were isolated from elk. They were called CWD1 and CWD2, and the main difference was that CWD1 had a short incubation period and CWD2 had a long incubation period. And all those studies are done in mouse models of prion disease. So you might now say, okay, a mouse does not have to do a lot with the deer, and I have colleagues that say a mouse is just a test tube with fur, which is true. I mean, there are many differences between mouse and cervix, but on the other hand, it's a cheap model and it allows us to study certain aspects of prion diseases, such as transmission and whether a certain species or the prion protein of a certain species would be able to refold if it comes in contact with the CWD prion. Then Debbie McKenzie's lab from the University of Alberta showed that passage of CWD prions between deer or between mice that express prion proteins of different prion protein genotypes, and I'll come back to that later, can generate new strains. And then there was also one publication that showed that strains are not dependent on the genotype of the deer where it comes from, but also they are dictated by the host species. So it would make a difference whether a deer becomes infected or an elk becomes infected. So a lot is not known about CWD strains, but again, it's critical to characterize those strains to be able to come up with an efficient diagnosis or prevention and to predict transmission to other species. So what makes the story more complicated, and now it comes back to the genotypes of the animals, is that in the cervid prion protein, there are so-called polymorphisms, which are variations in the composition of the prion protein. And each species, like mule deer, white-tailed deer, Rocky Mountain elk, they have specific positions or like amino acids that are the building blocks of the prion protein that can either be one or the other building blocks. So for example, in mule deer, it's 225, it can either be serine or phenylalanine. In white-tailed deer, we know 3 or 4 of such polymorphisms, and in the Rocky Mountain elk, I guess you're all familiar with the polymorphism at 132, which can encode either methionine or leucine. And what the effect is on CWD susceptibility, if people looked at the incubation times of disease in animals that express a variant of the prion protein, like here, in mule deer, the 225SF, the animals that express the non-wild type or not the most abundant prion protein have a long incubation period. Also in elk, the 132MM genotype is the most common one. It's the wild type, and they get disease earlier than animals that express 132LM or 132LL, and the same is true for white-tailed deer with variations at amino acid 95 and 96, and there are some examples in reindeer as well. So the common view was that animals that express variations of the prion protein have a longer incubation period of CWD, that they are less susceptible or maybe resistant against the disease. However, this view might be challenged a little bit by this study from Debbie McKenzie and also by work from my lab. So Debbie started out with brain homogenates from infected deer. And the infected deer, this was an experimental study, they expressed different prion proteins. So they expressed the wild type. Then she had one that had a variation at position 96. Then she had one that had a variation at amino acid 95. And then she had one animal that had variation at position 95 and 96. And she took brains of those animals and transmitted them to mouse models of CWD. And to a normal mouse expresses prion protein, but a normal mouse would not be, we cannot infect a normal mouse with CWD, so they are not susceptible to the disease. So we have to basically remove their own prion protein and then introduce the deer prion protein. And once they have the deer prion protein, they can be infected with CWD and infect the disease. And so we have those transgenic mice, which on one hand express the normal, so the wild type deer prion protein, and then we have mice that express the prion protein that has this variation at position 96. And actually this mouse model was thought to be resistant, so the people who created that mouse model, they have infected it with different CWD isolates coming from elk and coming from deer. And these mice did not develop disease with any of the CWD types they injected. So they gave the mice to Debbie and said, yeah, you can test them, but they won't get disease. they won't develop disease. So she transmitted all those deer materials into the two different mouse lines. And these graphs now show here on this axis the incubation time of disease. On the x-axis, they show which brain homogenate from which deer was used. And each dot here represents a single mouse. And what you can see is that all the mice that have the normal, the wild-type deer prion protein, they can be infected with all the different isolates. So all the mice came down at the longest, like 400 days post-infection. However, the mice that express that variation at position 96, if they were infected with the wild-type or the S96 wild-type CWD isolates, they did not come down, they did not develop disease, so they resisted this infection, which was kind of expected and which was told to Debbie. However, when she had material from the deer that had this variation at position 95, she could infect those animals. So this means that there are CWD variants or strains out that have the ability to infect animals that were previously thought to be resistant to infection. So from our own lab, we also started characterizing CWD isolates, and we received a lot of material from Trent Bollinger, and we found one that had a variation at position 116. It was also from a white-tailed deer. And we did a lot of biochemical characterization and also transmission studies, and we found also indications that this isolate contained a different strain of CWD than everything that was known before. So we also took material from the deer, transmitted it into a mouse model, and then we passaged it through the mouse model. This is a mouse model that expresses, again, the wild-type cervid prion protein. And what we found was that the mice that were infected with the 116AG deer material first had a longer incubation time than the ones that were infected with the wild type. But then if this mouse material was again transmitted into mice, we found that the prions adapted very nicely to those mice, so the incubation time became significantly shorter in 4 out of the 5 mice. However, one still had a much longer incubation time than the mice of the other groups. So we took now brain homogenates from these mice and brain homogenates from this mouse and transmitted it again in the third passage into those mice, and still we see the difference. So we have a strain probably that has a very long incubation period in that mouse, and then we have a strain that has a shorter incubation period in that mouse that expresses the normal wild-type prion protein. So what we want to take home from this is that the transmission studies we did confirm that there is a new strain, and in contrast to Debbie's study, where she only saw the difference in the strain when she injected mice that expressed a deer protein with a variation, we saw a new strain that was isolated in mice that expressed the normal prion protein. So we were working together with Debbie, so she was very interested in getting that a 116AG isolate, and she again transmitted or inoculated, infected her resistant mouse strain with this. And as with the H95 mice, again, these mice became sick when she injected them with the 116AG, which again confirms that there is a different strain. It is a strain that can infect a genotype that was thought to be resistant to disease before. So in summary, we saw that we could isolate a new strain upon passage in mice expressing wild-type deer PRP, and that also the 116AG prions cause disease in the so-called resistant S96 mice. So overall, it becomes more and more evident that new CWD strains can be isolated or can be found in animals that have those variations in the prion protein, that these new strains might potentially infect genotypes that are thought to be more resistant to the disease. And now the question is, which we would like to follow up, whether the transmission barrier of, so what is the transmission barrier of those strains to other animals like non-cervids, as I said before, sheep, cattle, and so on. So, as I said, it's all done in transgenic mouse models, but that's the best and cheapest way we can test such things. Of course, it would be better to do it in deer, to do it in elk, or whatever, but it's always you need the facilities and you need to do the funding for that, so that the mouse is really a model that lets us get hopefully as close as we can to the real disease. So another topic we are interested in is pre-mortem diagnosis of CWD. As you know, it's a problem. Prion diseases can only be diagnosed once the animals or the humans are dead, or so the current testing depends on the detection of the PRP scrapie, so of the protein that has changed its shape, and the materials that are used are either brain or lymphoid tissue. Then there are tests that are used for screening those materials, and then immunohistochemistry is done to confirm the non-negative or the positive results. In wild cervids, this depends a lot on hunter compliance, and all the tests that are used are done once the animal is dead, or if it's done on a living animal, it's invasive. You have to get those tissue biopsies, which doesn't make things easier. So what we thought would be needed would be a sensitive and specific preclinical detection in the animal that ideally can be done before the animal is dead and in a sample that we can easily obtain without having the animal in hand, without taking biopsy from the animal. And for the tests, so over the last 10 years there were many developments to make the detection of prions more sensitive. And one such assay was developed in the Rocky Mountain laboratories of the NIH by Byron Cowie, and it's called RT-QuIC, real-time quaking-induced conversion. And this test is way more sensitive than the tests that are currently used for detection of PRP scrapie, because it is based on the amplification of the PRP scrapie that is in a given sample. So you have a little bit of PRP scrapie, and then you subject it to the test and you amplify it to detectable levels. So this test allows to detect PRP scrapie as little as 1 femtogram, so 10 to the minus 15 grams. And it uses recombinant normal prion protein that is produced in bacteria as a substrate, and what is called seed is the sample from the prion disease suspect animals. And this can be like many different things like brain homogenates, cerebrospinal fluid, urine, and what we've used now was fecal samples. So this is how the test works. Here is the reaction plate. It has 96 wells, so we can test a larger number of samples at the same time. And we add to those wells the recombinant PRP. So this is the normal prion protein that is produced in bacteria. It is not the infectious form. Then we add our sample, which potentially contains the infectious form, the PRP scrapie. And then we add a fluorescence dye and put this plate into such a machine. It starts to shake the plate, and then the plate is not shaken, and it's incubated for 24 to 48 hours. And what happens during that time is that the PRP scrapie, so the seed that is contained in the sample from, say, the infected deer, would refold or would convert the normal recombinant prion protein into the abnormal scrapie form. And this happens over time. This is the amplification step. So you get more of those seeds, and then more PRP is converted, and this can then be detected with the help of this fluorescence dye, which binds to those protein aggregates. And this is what we can measure. So we can measure the fluorescence of that dye. And come up with such curves. So here on the y-axis, you see the level of the fluorescent signal, and on the x-axis, the reaction time of the RT-QuIC in hours. And once the sample turns positive, the fluorescence level increases. And then you can do different dilutions of your sample, and then which then has an effect on the reaction time when the sample becomes positive. So the increase in fluorescence indicates a positive sample. So these are results from my lab again. So we tested this test first with CWD positive brain homogeneity, and you see we did dilutions up to 2 times 10 to the minus 7. This red line indicates a cutoff, so every curve that is beyond that red line is a positive signal, and we can detect CWD prions in those brain homogenetes at a very high dilution. The same is true here, whereas in the CWD negative brain homogenate, we don't see the increase in fluorescence, so the test stays just at a flat line. So RT-QuIC has been tested for the detection of CWD prions in many different materials, and this was work done mostly by researchers from Colorado State University at Hoover and Kansas Tyson. And they've used saliva, urine, and blood to look for CWD prions. And we've also used urine performed, and what in all those studies was found that the infected deer start to shed very early. For example, in saliva, CWD prions was detected as early as 3 months post-infection. In urines, we had it about 6 months post-infection, and also in blood, it was about half a year after infecting the animals that it was detected in the blood. What also was interesting in the studies done by the people from the Colorado State University, they had deers of different genotypes. So they had deer that were thought to be more resistant to the disease, which have a longer incubation time, but they found that these deer also start to shed prions very early. Then a lot was done looking into ramal, rectal-anal mucosa-associated lymphoid tissue, so basically rectal biopsies and nasal brushings to detect CWD prions by RT-QuIC, and here elk samples from farm and free-ranging animals were used. And the detection in the rectal biopsies correlated very much with what was found in immunohistochemistry, and there was a very high specificity, which was a little bit worse for the nasal brushings. So it seems like the ramal also appears like a very interesting material for testing, but it would require to take biopsies from the animals. So we were thinking about using fecal samples, and I mean, the reasons are pretty obvious. Fecal samples are easy to collect, you don't need the actual animal. It's a real advantage for surveillance, or as an addition for surveillance in wild animals, because you're independent of hunters submitting their deer heads, and so on. And in earlier work, we have provided proof of principle that detection of CWD prions in feces is possible, but we only had samples from deer that were already in the clinical phase of the disease. And also, this is again a study from Colorado State University. They detected CWD prions in fecal samples, but these samples were also from deer that already were at the terminal stage of the disease. But our question now was, is preclinical detection positive? When would the animals start to shed? Can we find it before they develop disease? And to evaluate this idea, we worked together with Stephanie Schub from the CFIA Lethbridge, because she and Catherine Cranham, they have performed a study in elk where they orally inoculated elk with a single dose of CWD-infected brain homogenate. They had elk that had the wild-type genotype, the 132MM, and then they had a few animals in that group that had the 132ML polymorphisms, so a genotype that is thought to be less resistant to the disease. So the animals were inoculated and then they were euthanized at defined time points after the oral infection. And then the CWD status was confirmed by immunohistochemistry and Western blotting in lymph tissue to show that they were really infected. The fecal samples, they were not collected from the sheds. They were collected during necropsy directly from the rectum. So we used the negative samples, we tweaked our reaction conditions a little bit, so we concentrated and purified the PRPs greatly, because one main problem of that technique is that the bacterial or the substrate PRP, the normal PRP we put in, can convert spontaneously. So if you have the wrong reaction conditions, you might end up with such curves. So this is from a CWD negative animal, but we still see a positive signal. So we tried to adjust our assay conditions and retested the negative samples. And so by doing this, we brought down the signals to the negative baseline. And then we looked at the positive or the fecal samples that came from the CWD infected animals, and we found something. So we found slightly positive signals at the very end of the reaction. So we were thinking of ways how to improve the detection, how to increase the sensitivity of our test. And to do this, we added, after 25 hours of the reaction, we added fresh, normal substrate prion protein, and then let run the reaction for another 50 hours. And by doing this, we could increase the sensitivity. So this was the sample before adding new substrate, and this is the sample signal if we add a fresh substrate. So we could increase the specificity and the sensitivity in those experimental samples. So this is the outcome. You already see we didn't have many animals. So again, these were elk. They are kept in a high biosafety level facility. It's very expensive to keep those animals. So for each time point, we only had a limited number of animals. So here is the genotype of the animals, and this is the time point when they were euthanized after infection, so the days post-infection. And after 8, 9, 14 days, we see something positive in the fecal samples. However, it's not very reliable because it might just be what goes from, I mean, you put in a large amount of prions orally into those animals, and you would expect that it takes some time until it goes through the gut and comes out on the other end of the animal. So we wouldn't know this, but after 42 days post-infection, we also saw like positive signals, and at that time, everything that went in 40 days to before should have come out of the animals. So we think that the animals start to shed very early, and here after 56 days, both animals became positive. And the other thing we found, if you look here at this animal with the genotype LM, it also had a positive signal already after 150 days post-infection, which is very early compared to the extension of the incubation time they have. We had a few animals that developed clinical disease. 3 of them were positive. One again was an LM animal, and this was negative. So what we overall would say, we can use RT-QuIC for detection of CWD prions in a wide variety of specimens. The detection in fecal samples is possible at early time points after experimental inoculation. The elk with the 132 ml genotype, they also start shedding at early preclinical stages, which confirms the other studies where they used deer material and looked in saliva and blood, and also found that the potentially more resistant animals start to shed early. And what we're now doing is we collect samples from everywhere, and this is where I would like to ask for your help, because we're always in need of negative samples to validate our assay. So we received samples from that were collected just from the forest in areas where there is no CWD yet, and we received samples from the hunter station in Wainwright in Alberta, where the hunters go and submit their heads where the test samples are taken. We will receive samples from Catherine, from the areas where the infected deer overlap with the caribou. We received samples from depopulated elk and deer farms, both from Saskatchewan and Alberta. And the idea behind that is to see how applicable our tests that seem to work in experimental conditions where the animals are kept in a defined environment, they get their defined food, so what comes out, like the fecal composition is probably very similar between the animals, whereas if you have wild animals, they have different food, and this might affect our test sensitivity. So we just need lots of negative samples to validate our protocol. And here, coming back to the remote samples, I just would like to mention briefly a study that's coordinated by Dr. Nick Haley, and I think you know him. He spoke last year at this occasion, and this was a cross-validation study, also using RT-QuIC, but using elk rectal biopsy samples. So cross-validation means, so Nick collected samples from an elk herd in Colorado in 2 subsequent years, and he sent those samples to 5 different laboratories for testing by RT-QuIC to see how the test variation is if different labs perform the assay. So this was a blinded study, which means we got the samples, we didn't know which ones were positive and which ones were negative. And another aim was also to compare results obtained by immunohistochemistry and by the RT-QuIC testing. So we got a very defined protocol from him, how we should run the assay. We had in total 700 rectal biopsies. Each lab made its own recombinant PRP substrate, so this is a variation, and each sample was tested in triplicate. And if all 3 of those samples were positive, the sample was considered positive. If 2 were positive, the animal was considered positive, and if only 1 was positive, we called it suspicious. And this was what came out in year 1. Out of 300 or almost 400 samples, we had 58 positives, 10 suspicious, and the rest was negative. In year 2, almost the same number of samples, again almost the same number of positive samples. What was much higher was the number of suspicious samples. So this could be animals that became infected over that last year. And what was also interesting was that there were samples in the first year that from animals that were suspicious, and they were negative in immunohistochemistry from the same material, but when they came back in the second year, they were positive in RT-QuIC, and they were also positive in immunohistochemistry, showing that the RT-QuIC indeed is more sensitive and allows us earlier detection of CWD prions in the rectal biopsy. Yeah, and this is just another summary showing that from year 1 to year 2, mainly the number of the suspicious samples increases, but also the overall number of the positive samples from 14 to about 16%, which just confirms that the disease is spreading among those herd. So this study will be continued with 2018 samples, so he is, I think, around this time now, he'll be there and collect more samples from those animals. Okay, and now coming to the last part, and I hope you still bear with me, and this is about an approach to prevent or treat CWD. And this is not about vaccination. This is about using chemical substances to treat the animals with. And it basically goes back to a publication from a Japanese group where they used where they used chemicals called cellulose ethers to treat mice and hamsters that were infected with prions. And cellulose ethers are pretty simple molecules. They are soluble in water. They cannot be digested by our body, so basically they are sugar molecules, polysaccharide derivatives that come from plants, and our body cannot digest them. They are already used very much. They are inactive ingredients in food, in cosmetics, and in pharmaceuticals, so there seems to be a low risk associated with taking those drugs. And what that group shows, again, this is done in a hamster model of scrapie prion disease. So they infected the hamsters intraperitoneally, so not directly in the brain, but into the belly. And this should basically reflect what happens if we acquire prions orally. So the prions have to go from outside of the brain, they have to make their way into the brain and then start the disease. In non-treated animals, this took about 100 days. Again, this is the incubation period of the disease, and the gray line were the non-treated animals. However, if they gave the animals a single injection of that cellulose ether into the skin or subcutaneously, 3 days after the infection, they found this very large increase in incubation time, which was from 97 days in the non-treated animals, and then extended by about 600 days in the treated animals. So they tested other applications of the drug. So here it's the same way of infection. It's into the peritoneum of the animals, and then they started treating them 7 days after infection with a 15% cellulose ether diet. So they fed the cellulose ethers until the animals came down with the disease, and even this resulted in an extension of the incubation time of 200 days. And what is most interesting maybe is that the drug was also effective if it was injected only once, more than a year before the prion infection. So here they gave the animals one injection of the cellulose ether. Again, it was subcutaneously into the skin, and after 13 months they infected them with prions, again into the peritoneum. And this treatment resulted in a twofold increase in the incubation time. So you give a drug a year before prion infection, and then a single application, and then you infect them with prions. And the treated animals would have an incubation time twice as long as the non-infected animals. So the problem with this study was that it seemed to depend on the prion strain that was used for infection. So I told you before, the prion strain can affect the treatment efficiency. So we cannot necessarily say this works for all prions or it works for CWD prions. So what we are currently looking at, can we use this to treat CWD infection, again, in our mouse models. First, we used RT-QuIC, so our in vitro prion amplification assay, and added the cellulose ether to the RT-QuIC reaction. We had CWD isolates from elk, from mule deer, from white-tailed deer, and then our 116AG white-tailed deer. And in all those reactions, the addition of this drug could completely inhibit the amplification of prions, whereas in the untreated reactions, you see a good amplification of the prions. Then we did mouse bioassays, so we had mice that express elk PRPC. We used elk brain homogenate for infection, and we had a group of mice that were not treated. And then we had two groups of mice that were treated with one dose of that cellulose ether, either at the day of infection or 1 month before infection. And infection, in this case, we injected the prions directly in the brain, which is the most direct and the most efficient version of prion infection. And in this curve, again, this is the survival time. Each dot represents the survival time of a single mouse. This is the control group. They all died or came down with the disease at around 95 days. And then the treated animals, they had a strongly extended incubation time of about 20%. And actually we saw a more efficient extension of the incubation time when the animals were treated 4 weeks before the prion infection. So we injected the CE 4 weeks before prion infection, and we get a better effect as if we inject them with the drug at the same day of prion infection. So in summary, we show that in principle the cellulose ethers seem to have an effect on CWD amplification in vitro, and we also see that the treatment strongly extend the incubation period of CWD in transgenic elk. What we are now doing, we're testing other cellulose ethers. We use different CWD isolates for inoculation. We want to do the peripheral, so the intraperitoneal infection of the mice. But again, we extend the incubation period, and it's always a critical question with CWD. If you extend the incubation period, would we affect the shedding? So what is the value of any drug that extends the incubation period of a genotype that has a longer incubation period if we cannot reduce the shedding of the prions? Because then we might just end up with animals that live longer but shed prions twice as long as the non-treated animals would do. So overall, I said the limitation is it's done in mice, but I would be curious to hear your opinion about this, because I think this would be a method where, especially with the farm elk, you handle your animals. You could give them one injection with that drug at a certain point, and maybe they would be protected for a longer time. So it's nothing you have to keep. You have to treat them continuously. It's not a vaccination. It would be just injecting them with a drug that potentially extends the lifetime, or hopefully we find modifications of that drug that even prevent the onset of the disease. Yeah, that was all I had to say. And I would like to thank my group, my collaborators, funding agency, and I would like to bring your attention to this Genome Canada, Genome Alberta, and Alberta Prion Research Institute funded research. So this is a large project that will go on over 4 years. It's a collaboration of 12 groups that work on prion research. We look at different aspects. We look at strain characterization. We look into diagnostic assays, and not only RT-QuIC, but also other looking for other markers, potentially in fecal samples, and so on. And this group also, they always look out very much for all kinds of samples, like blood samples, tissue samples, and we already got a lot from depopulation with the help of Gordon Mitchell from CFIA. Catherine Neal is also a person who provides samples for that study, and so on. But this is also another opportunity to get involved or to help us with our research. Yeah, and last but not least, so this is, you might hear I have an accent, I was asked already, so I'm from Germany. And this is my brother-in-law, he has red deer, so he's farming red deer. And these are some pictures from his animals I took 2 years ago in June when we were visiting. So the whole deer farming, I think, is very different in Europe than it is here. So they keep the animals for meat and for to sell those with the nice racks for trophy hunting. They don't do the velvet stuff, so they don't harvest the antlers. Yeah, and they are small. I always thought red deer are small, but until I've seen elk, which are really tall. Yeah. Thanks for your attention, and I'm ready for questions.
You want to connect that link to our website so that we can access that on demand if we want to, rather than go fish around and try to find a link. But the other thing, with your permission, can we distribute your PowerPoints, your presentation
To as a, you know, if it's scientists, then I'm hesitant, but I think here, yeah, it's this one. Yeah.
Okay, so if anybody wants the slides of the PowerPoint, we can get those sent to as well. So anyway, having said that, I'll open the floor for questions. Blake looks like he's got a mic.
So in using the RT-QuIC as validation for products in antler harvest, if we collected a fecal sample from an animal at the time of harvesting the antler, and then had that fecal sample tested, what kind of percentage could we guarantee at this point with what you know that that animal is a safe?
That is a good question. I think it's a very important question. The problem we have now is that, I mean, you saw the sample numbers we had from the experimental animals. It's very limited. We did not go into quantification of prion amounts. So we do not know a lot about sensitivity, and the problem we have now is that we cannot confirm whether a sample that is positive in RT-QuIC, whether it's really positive in terms of it contains infectious prions. So at the moment I'm very cautious about making any predictions because with our work that we continued now using those wild samples, using those samples from wild animals and from hunter harvests and from depopulations and so on, we saw that unfortunately, especially in the deer, the assay does not seem to work as good as it worked for the elk. So we see a lot more of those false positives, which is a problem. So we have to go back again and tweak our assay to reduce those false positives. For elk, it seemed to work very reasonably. So I have one example. We received elk fecal samples from a depopulation. They were tested at the CFIA, and 2 animals turned out positive out of 70. We have tested the fecal samples of 35 of those animals, and we found 2 positives as well. So with the elk, it seems to be more reasonable, but I think it needs a lot more validation, in particular negative samples, to really say if we have a positive signal, we can say this is true positive, like with a 95% probability.
Okay, so how would we, I'm involved in marketing the antler in both processed form as well as green form, and we need something, and we only collect antler from elk. It's not collected from the deer.
Mm-hmm.
Okay. So how do we go about expediting this process, contracting a lab, private research, whatever, or is this something that you could help us with, or how do we go about doing this?
I mean, if you're talking about antler, probably the better way would be to directly test the antler velvet, right? Or
okay,
like the
so if we pulled a blood sample from the animal, because antler is basically a blood product, that's what we're harvesting.
Mm-hmm.
So if we pulled a blood sample from them, which is very easy to do during the velveting process, could that be used now?
Yeah, I mean, every sample, I think at the moment, the most, or in my impression, the most reliable samples are really the rectal biopsies. So for those samples, there is the best correlation between immunohistochemistry, between other tests. I think the highest number of animals have, or like samples have been tested for the rectal biopsies. So if if you would ask me what tissue I would rely most on, I would say the rectal biopsies.
Okay, so even to start, if we were concerned about a particular processed product or something that we wanted to verify was safe as much as we possibly could, a rectal biopsy at the time of antler harvest would be the way to go?
I think it would give you a good indication.
Okay.
Yeah.
Great, thank you.
But again, I mean, it's always, there's always a window when the animal got infected until it becomes detectable also in the rectal biopsies. But I think it's before the immunohistochemistry becomes positive. And yeah,
So if we collected the sample 40 days later,
I mean, yeah, this could be one way. So
and or
like
harvest,
you harvest, you have a sample or you have a rectal biopsy, you send it in for testing, and then maybe using what I described, we do it in triplicate, and we have a suspicious sample. So one way could be to go back to this animal like a month later or 2 months later and take another biopsy
Right.
and test and see whether it became more positive.
well, if we even had a question, we could identify and tag each piece of that, or so we could eliminate that animal from the process.
Yeah, or at least
Yeah.
for a window until there will be retesting.
Okay, great, thank you.
I'm gonna follow that up with a question, because it leads right into that, is how many numbers of samples of animals are we gonna need to have in order to say, okay, we're close to validating this test, or like what you said, you've got 16 samples or whatever the number was, and you come up with this result. If we come back and we say we have 1,500 samples, is that the right number? Is 5,000 the right number? What is the right number? Because I think what Blaine's alluding to here is that we want to get this process going, so how many numbers do we need?
I mean, in the end, I would say the more the better, but the problem is that in my lab we have limitations too, so it would be better to get other labs on board as well. Because I'm not in that sense a diagnostic lab that I can do high throughput testing, like we cannot process thousands of samples. So my numbers are like limited to 50 to
I
100.
understand that.
And I'm-
But scientifically, how many samples do we need as a control? Ballpark, obviously, we're not going to hold you to this, but we've got to have some reference to start, because we're coming into velvet harvest. We can probably get enough guys to collect fecal samples and possibly rectal biopsies as well, if you want samples. And contract a private lab, whatever it takes. There's a misunderstanding, especially in the wildlife community, that our velvet antler and we're not making money. We've had a 600% increase in the value of velvet antler, the raw product, in the last 4 years, and it's steady. It's stayed. The product is in demand. The processed product is gaining traction. I need to be able to tell the consumer and guarantee that it's safe.
No, I totally understand that, but it's
I think Catherine wants to answer to that question, please.
I just want to clarify that from the wildlife perspective, it's not about making money. I understand there's a lot at stake here, and that's what I was speaking to, is we need to address the risks that are here. It's not about money, it's about what's on the landscape and how do we address it.
No, but for us, it is about money.
But the wildlife, when you say the wildlife community, I want to be clear that that is not an issue that I've heard. Whether it's something you've heard
Well,
that
I get it all the time from the wildlife people. You guys aren't making money, there's no way you can make money, you'll never make money, period.
Concerns have been raised is the CWD on the landscape, and how do we answer this question?
I understand.
Sabine, you've got a couple of elk ranches in your backyard. With you being at the U of C, just south of Calgary, there's a couple of elk ranches. And like Blaine has alluded to, we are coming into velvet antler season, and we very easily can start drawing blood samples and taking fecal samples and bringing them into you. I mean, we're close, literally close.
Yeah. Yeah, so definitely, I mean, we didn't do any work on blood, so, but we would definitely be interested in the fecal samples, but with the limitation, it's for research, I mean, it's research use.
That's right.
We do
But
have
we
to
have to start somewhere.
Yeah, that's clear. So I mean, as I said, samples for validation is always good, but don't expect too much from us. Like even if we're testing and we see they're negative, I think it takes much more than just having a research lab, establishing a test and testing a few samples to really validate it as reliable diagnostic test, I think it takes much more. Things like that cross-validation that has been done, like repeating lots of samples, comparing it to results with other tests that we're not doing. But I mean, of course, you're right. We have to start somewhere. And if we get to a point where we can say, okay, we have samples that come from negative areas, from negative animals, and we get them out negative in our test, I think that would already be a success.
Thank you very much.
Ian?
Do I need a mic?
You probably did.
I've got the volume, no problem. Didn't know whether you wanted me to use the mic.
Use it because then it'll get picked up for the recording.
I have about 30 questions, but we're not going to be able to do all that right now. Yeah. Very specifically, the next step with Ramold, in particular, because you recommended it as being the most accurate to date, and RT-QuIC, which appears to be the most accurate to date. What path do we need to follow with what lab to validate and get it running on a commercial basis? And I'm going to target PDS because we're working with them now. And who would train them to do it?
I mean, if it comes for the trainings, we had people approaching us from the Alberta government as well, from the people who do the agriculture division, who do the testing for the farm deer. And so, like, training people in our lab, if you send one or two, that would be fine with us. I think to get a test really approved, it probably would need input from the regulators.
You said this to me.
Yeah, maybe we can talk about that.
Penny likes you, so that's a good start.
Okay, so we need to start that process, so we'll talk more about that, but that's definitely a high priority.
Yeah, and just to add, I mean, this was not my study. It actually was Nick Kelly who started all this, so it would be a good idea to talk to him as well about this. We were just, helping validate his essay and using his essay to show that if one lab or the other does it, we come up with the same results.
I still have my heartbeat.
Remind everybody else up behind.
The red deer, okay, and you didn't mention the red deer as being acceptable, but then you mentioned there have been red deer positives in Norway, which I've never heard of before. Have you ever heard of the red deer positive in Minneapolis, Minnesota? That was about 4 years ago, and it was a herd of red deer on a little heritage farm in the middle of Minneapolis, owned by farms, and they had a red deer positive there. We never want to forget to mention red deer because the New Zealanders are always holding their heads so hard and high and saying, we're above this, but they are susceptible.
Yes, they are. I mean, that's been known before, but now in Norway, like, they have those wild animals. Yeah.
Thank you. More later.
You have to surrender with Graham, up where two.
Hi, Graham Mansfield, the Ministry of Agriculture. I just have two actually kind of separate questions. One's more RT-QuIC and one is more general. So at the beginning, when you're doing the overview of all of the kind of areas of the animal tissues, etc., that prion proteins can be found, I may have missed it. There's one thing that's generally overlooked when people are doing that summary that I would like you to comment on. Is the potential for presence in germplasm and transmission by artificial insemination or embryo transfer. And then secondly, separate from that, we talked a lot about strain differences. So I'm interested in what that looks like on the other side of RT-QuIC. If we've got the same substrate going in, we've got differences in tertiary conformation and presumably under the same experimental conditions are going to have differences in outcome. I'm just not clear on how that interaction between the strains is looked at and whether or not we can even directly compare results from different strains because of those potential differences.
Yeah, that's an important question. So to the first one, I mean, it's known that CWD can be transmitted from mother to offspring in utero. I cannot comment on in vitro fertilization in insemination because I'm not aware of any studies that have been done in that respect. To your second part with the strains, yes, it could be a problem. So different strains can react differently with with one given substrate. And so what we usually do is we try to use different substrates, but we haven't observed with any of the isolates we are working with that it does not amplify at all with what we are using. But for example, for BSE or for human prion diseases, there are substrates that don't work with certain types or certain strains of prion. So that's really valid questions, and it just shows again that we have to really be careful with strains and everything.
Back to the cellulose ethers. Do you guys know why it's having an effect? Does it block CWD, or is it bonding with the protein, preventing it from the body absorbing it? Or how did you even stumble onto this? Or maybe you didn't stumble on it, but how did you get to this point?
So actually the original group, it was a little bit stumbling about it. So they tested other drugs, and they had drug preparations that had the ingredient they thought that would help against prion disease, and then they had their control without the ingredient. And they found that the control, even here with the controls, they even had extended incubation times. So they were looking at what's in the control, and they found this cellulose ethers. So how it exactly works, what we know from the RT-QuIC, the in vitro amplification studies, it seems to block the conversion of the normal into the pathological prion protein. But how it affects in the body, so those drugs, they're soluble in water, but then they become pretty viscose. So it might be that they just separate the different proteins from each other in a way that they cannot interact anymore, so that the normal form cannot interact with the pathological form anymore, and thereby prevent its conversion or its refolding. But what exactly happens in the body, we don't know. Thank you.
Any other questions, Lance, from behind here? No. Russell?
Do you know if there's any studies or birds to be created for like an allergens?
Sorry, can you repeat?
So like an LL is the resistive gene. Is there any birds being created to that extent? So then you do all the studies on pure LLs?
No, I'm not aware. I mean, there are probably you'll hear in the talk later about selecting resistant genotypes by breeding, but I'm not aware of an experimental study that used
I think the answer to your question, Russell, is that there hasn't been enough LLs concentrated in any one place at one time to be able to do that kind of work, that kind of a study.
Because there is LLs.
There is, yes, but it hasn't, those herds haven't been studied. I think that's what the...
Isn't there a herd in Colorado that's straight out?
You'll hear about him
He's
later.
here to talk about it.
I'm more interested in the genetic resistance of, and I guess my question is you've had scrapies around since the 14th century, and it looked like a pretty easy solve when they, looked like they bred RR or AA sheep, and in one cross, you've got 100% complete genetic resistance to scrapie. So I guess then the question would be, with all your studies and all these varying, continually morphing strains of CWD, I'd like to see more work done on, looking for the quote, silver magic bullet in terms of, finding the genetic resistance, especially in the elk industry. I mean, there's other guys that are here with the deer as well, but it seems when you're working on your mouse projects all the time, every time you think you come up with something that's a little more resistant, it seems like there's a cross reaction because the CWD strains keep morphing.
Mm-hmm.
What's your opinion? Are you really focusing on that, looking for that genetic resistance marker that we can use?
Mm-hmm. No, actually we are not looking at the genetic resistance marker. The only thing why I'm saying that, in my opinion, there is no complete genetic resistance to prion disease. I mean, if you look at all those polymorphisms, and this is only about the prion protein, if you have those variations, the animals have an increased, a longer incubation period, but eventually they would also develop disease. In scrapie, it was a little bit the same. So there is the classical scrapie, the one that's been known for a very long time, where the resistant genotypes were bred. But then there is also that atypical scrapie, and the atypical scrapie also goes into the sheep genotypes that are resistant to the classical scrapie. So it's the same play. I think the difference with scrapie is, again, it does not involve white animals. It's about a much more controllable system.
But if they were able to do it in scrapies, solve the problem, are we going to be able to do it with the cervid species? Do you really think if we keep looking?
At the moment, from all what I see about prion protein genotype, there might be other genetic markers that we are not aware of at the moment. But from all what I see with the variations in the prion protein, I would be hesitant to say that this would completely eliminate CWD. Because at a certain point, they all become sick, even if it was 2 years later than the wild-type animal. But maybe we get a different insight
I think what
afterwards.
Bruce is fishing for here is that, are we going to create a genetically modified cervid, which would be a GMC, to put at the top of the curve, of course.
Well, I don't care. I'll be blunt. I'll take a GMC. If they're completely resistant to all strains and forms of CWD, I'll take it.
I mean, if you want that, then it would be easy. Then we remove the prion protein gene. We create PRP knockout cervids. Huh? It was done with cows. Yeah, it was done with sheep and goats. But the question is, I mean, the protein has a function, and you don't know whether you do any good. And you, of course, would prevent the disease, but it's something that people are really hesitant to.
We're going to take one more question, and then we'll elaborate. Catherine, get louder.
I'm just curious from the group, because my concern was, how do we solve the problem? If solving the problem, I'd like to hear from you, and you don't have to say it out loud here, we can talk about it later. But what I want to know is, if you create a resistant animal, and as Sabine mentioned, it continues to contaminate the environment, have we solved a problem? And that's what I'd like to leave with you. Have we solved a problem? How do we get there? And what is the solution? Are we only interested behind the fence, or are we interested on both sides?
Well, remember, in scrapies and the papers that you read so far, if you've bred that genetic resistance, they don't get scrapies, and if they don't get scrapies, they don't shed prions.
And I've also heard from a researcher that they're not testing for scrapies as much as like sometimes with BSC, it's not testing for.
Well, I think you hear from the sheep industry, they think they've solved the problem because of their bred up genetic resistance now.
I'm going to leave that to Sabine.
Yeah, no, I mean, as I said, from what is out in the literature, the potentially resistant genotypes in deer or elk is that they have a very extended incubation time, but are they really completely resistant? They develop disease, they have prions, they shed prions. And maybe that's different to the sheep, right?
Well, no, we're aware of that. I mean, they found the magic 179 codon that they focused in on, and if they bred it, they had 100% complete resistance. We're aware that every once in a while an LL will come down positive, and we don't think I don't think the researchers have found the magic bullet yet. I don't know what marker it is. And if you have these morphing strains of CWD that keep cross-challenging, I'm not sure it's possible, but I think you can test for the disease till you turn purple. But if you can build an animal that's genetically resistant to all forms of CWD, of course, that's the magic bullet.
Yeah, no, I mean, of course, preventing the disease would be, and yeah, that's what we all want, be it wild or farmed animals, I guess.
I'd like to add to that the sheep industry is an industry that has been around, like you said, forever, and they've had scrapies forever, and it is still a growing industry that is still valid today. So they are living with it. It is in the food industry. It is one of the #1 eaten meat in the world. So I think that's a positive that when you keep bringing up the sheep, that industry still exists today. And if you look at how sheep are farmed, they're farmed in close quarters in huge numbers. So if you're, I think that you can draw some positives out of that from what you're saying there, that there is something that it can be done. I think that, in my opinion anyway. Another thing, a question that I have for you, you referred to 4 different strains of CWD in the deer, right?
Mm-hmm.
And you referred to a wild strain in your testing. Am I right, or did I misread that?
So what I was saying is that this strain, this 116 AG, it came from a wild animal
Okay.
that was from Saskatchewan, that was found dead close to a farm. And so we were just lucky that when we asked Trent for sending us CWD positive brains, that we got that one.
Okay.
Yeah.
So there's been no testing then to see if that is the same strain in other wild deer, or?
No.
But that would be an interesting thing to find out, that if that wild strain is actually its own thing. I mean, I just thought that was something
Yeah.
that would be interesting in the future
Yeah.
to see if they could compare that.
No, I mean that's the problem. That's what we want to reflect in our mouse transmissions. Like you have prions that come from a deer with a certain PRP genotype, and then we put it into mice that have the wild type, which is like the likely situation, at least in the wild, that there is that one animal that has a rare variant of the prion protein, it gets CWD, and then it transmits back into an animal that has the wild type prion protein. But of course, as I said, we cannot. The mouse models are helpful, but they are also to a certain point artificial. So all we create in a mouse model in the end would need to be confirmed or seen in deer models as well. And this is, I think, something that would be nice, but it's not really possible, at least for us.
Yeah, I just want to make
One last question.
I have one more quick question. But, at the start of your presentation, when we were talking about CWD showing up in Norway and not really knowing how that it showed up there, and that there was 2 different strains there again, you had said that it's possible that it could have occurred spontaneous, but there'd be no way of really proving that it was spontaneous or not. But we have to assume that all forms of the prion had to be spontaneous at some point. Right?
Most likely. So I mean, what I know from, the diseases can be sporadic in humans, it can be spontaneous in humans, so I don't see an actual reason why it could not be spontaneous in a deer or in a sheep or in a cow.
Okay,
I just want to clarify the point on strain, just so people don't get a strange view of the actual statistic. There were more positive scabies in Canada last year than there were positive cervids for CWD. So scabies still occurs in the sheep industry. They're not all resistant, but that's possibly because not all people are raising those resistant animals, and they're still accepting the possibility, the risk of scrapie, and CFIA still depops and compensates scrapie positive sheep herds.
Okay, no, sorry, we've got I know it's good that we've got to kind of keep things on schedule. We're going to have a little break, sorry, first. We've got an open session later with all the speakers, so with that, we'll take a 15-minute break and get everybody started back up again for the next presentation.